Welcome back to this educational channel on biology, everyone. Today we will continue to discuss the Pahang Bio Paper 2, and we will discuss some other structured questions, which are good questions to practice. So I hope you have already downloaded the Pahang State Paper, Bio Paper, from websites like SPM Trial Papers, right? And that you have attempted to answer the questions yourself. Okay, so you need to practice by training your brain to answer the questions and recalling the facts. Then after that, take out a red ink pen and mark the paper, and as you view this video. After that, the last step is to memorize the answers given and also write it down into your question paper. Okay, memorize the marking scheme. So that will benefit you a lot, all right, and prepare you for the SPM. Along the way, we'll also learn to apply some of the acronyms that I have invented, all right, and have discussed in my teaching videos. And you'll find that using these acronyms make it much easier for you to answer the questions, especially acronyms on respiration and fermentation. All right, so without further delay, let's get started. Question 1 is based on Form 4 Chapter 2, Cell Biology and Organization. Diagram 1 shows the structure of an animal cell. On diagram 1, label X in the space provided, right? So label X here in the space provided here and not below here. If you write below here, you won't get your mark, okay? So follow the instructions. So what is X? Now, if you were to enlarge this X here, it should look something like that, right? There's a folded inner membrane, all right? So this is a special characteristic of what organelle? Yes, the mitochondrion, right? And since only one mitochondrion is labeled here, it should be mitochondrion and not mitochondria. So mitochondrion is the best answer. Next, draw an arrow to match component Y to its function. Okay, now this is component Y. So Y is a vesicle that is found in between the rough endoplasmic reticulum here and this structure called the Golgi apparatus. Okay, to recognize the Golgi apparatus, we can see that it has a parallel C-shaped structures like that, and then it forms its secretory vesicles also. All right, so this is the Golgi apparatus. Now, so Y is found in between rough ER and Golgi apparatus. Therefore, it must be what vesicle? Transport vesicle. Okay, it must be a transport vesicle. So what is the function of this transport vesicle? Yes, it is to transport proteins synthesized by ribosomes from rough endoplasmic reticulum here to the Golgi apparatus. Okay, so the transport vesicle will contain the proteins synthesized by ribosomes. So at first, ribosomes synthesize proteins, and then the proteins move into the space in the rough endoplasmic reticulum where it's transported until it reaches the end here, and then the rough ER will enclose the proteins in the transport vesicles to send them to the Golgi apparatus. All right. So transport vesicles transport synthesized proteins or proteins synthesized by ribosomes from rough endoplasmic reticulum here to the Golgi apparatus. Right. Now, is this second choice correct or not? Transports modified proteins. No, because the protein in Y is not modified protein yet. It has to be transported to Golgi apparatus to be modified. Okay, so it is not modified yet. So this is not the right answer. Anyway, what is the structure that transports the modified proteins? Yes, it is the secretory vesicles. Okay, so after Golgi apparatus has modified the proteins, the Golgi apparatus will enclose these proteins in the secretory vesicles, okay, which transports the modified proteins to the plasma membrane and then secrete the proteins out of the cell. The modified proteins are like enzymes, for example, okay. So transports modified protein is the function of secretory vesicle. And lastly, synthesizes proteins. What is the organelle that synthesizes the proteins? It's the ribosomes. Question C, explain one characteristic of X. X is the mitochondrion. So looking at the structure of X, we can see that it has a folded inner membrane. The folds are called cristae, or singular form is crista, right? Now what is the function of these folds? So it increases the total surface area, right, of the inner membrane. Without the folds, then the inner membrane will be a smooth structure with less surface area, okay? But the folds will increase the total surface area. For what purpose? Now we must be aware that the reactions occur on the surface of these folds, all right? Reactions that are involved in cellular respiration, okay? So the more the total surface area of these of the inner membrane, then the more reactions that will occur on its surface, okay? So that more energy or ATP is produced through cellular respiration. Okay, let's see your answer. Now every point here is 1 mark, P1, P2, P3, 1 mark each. It has a folded inner membrane, meaning the mitochondrion or X has a folded inner membrane. To increase the total surface area, this causes more energy to be generated through cellular respiration. Okay? Now, if you can mention the word cellular respiration, is also important as part of the process, as the process that occurs in the mitochondrion. Now, don't mention that the mitochondrion has a folded inner membrane because they are referring to X. All right? So your answer must have the word, the letter X. Okay? And not the name of X. Now, next one, justify why ribosome is attached to the rough endoplasmic reticulum. Justify means to state or explain or prove that something is, why something is true or something is like that. So why is it that ribosome is attached to rough endoplasmic reticulum, right? So you have to explain the functions of ribosome, rough endoplasmic reticulum, and so forth. So actually, the ribosome is attached to the rough endoplasmic reticulum here, all right, so that it can, so that the proteins that is synthesized by it can move into the space in the rough endoplasmic reticulum and be transported by it, right? And after that, the proteins, the rough ER will enclose the proteins in the transport vesicles and send them to the Golgi apparatus so that they can be modified. Okay, that's the reason why the ribosome is attached to the rough ER. It's near to the transport system because it needs to have its proteins transported to Golgi apparatus to be modified. So let's look at the points. Ribosomes synthesize proteins. Secondly, these proteins then need to be transported by the rough endoplasmic reticulum to the Golgi apparatus. Okay, so that's why the ribosome is attached to the rough endoplasmic reticulum. And you need to send the proteins are transported to Golgi apparatus to be modified so that a specific enzyme or hormone can be formed. Okay, so any 2 points here, you get your 2 marks. Same for question C. So here in actually question D, you are explaining the functions of the ribosomes, the rough endoplasmic reticulum, and the Golgi apparatus. So this is a good question. So these 2 questions are like hot questions, and it's quite good to remember how to answer them. It's quite important. Question 2 is based on Form 5 Chapter 1 contents, Organization of Plant Tissues and Growth. Let's read the question. Diagram 2.1 shows the growth curve of a plant that lives in temperate climates. All right, and you have to identify the types of plants that show the growth curve as shown in diagram 2.1. All right, so analyze the graph first. All right, it shows the height against the time in years. You can see that this graph consists of 5 sigmoid curves. 1, 2, 3, 4, 5. Each sigmoid curve represents 1 year of growth. Okay, starting from spring, then to summer, when the growth is fast, and then late summer, autumn, and winter, the growth is slow. Okay, and then after that there's a new year again that starts off, and there's rapid growth and then slow growth again. Okay, so but the plant is more than 2 years. So what is the type of plant that shows this kind of growth curve? It must be a plant that lives more than 2 years. Okay, and the answer is perennial plant. So what's a perennial plant? Now, perennial plants are plants that live more than 2 years. They have a longer lifespan than the annual and biennial plants. Annual plants live for 1 year. The word annual means 1 year. Biennial means 2 years. Okay, a plant that lives for 2 years. Okay, but these perennial plants, they are more fortunate. They can live for a longer lifespan, which is more than 2 years. Okay, and they can be classified into 2 categories: the woody perennial plants, like the fruit trees, okay, like rambutan tree, mango tree, durian tree, and so forth. And the herbaceous perennial plants are those that don't have wood in them, but they can still live longer than 2 years. Okay, they are smaller in size than the woody perennial plants. Right, next question. Give 2 examples of plants that have a growth curve as shown in diagram 2.1. That means 2 examples of perennial plants. Okay, what is your answer? Now, the most common one that we know of is hibiscus plant. Like in my school, I have there's a hibiscus plant that has lived for many years since the new school was built. I think at least 10 years like that. Okay, and then what about the another example? Okay, you can give examples of fruit trees, the big fruit trees, such as mangosteen tree, mango tree, favorite is durian tree, jackfruit tree, and so forth. Any one. So any one of these examples can be used for number 1 and 2. Okay. Next question. Diagram 2.2 shows a cross section of a tree taken from the temperate areas. Based on this diagram, estimate the age of the tree. So if you look at this diagram, it is the cross section of the stem, woody stem of the tree, and you can see a lot of gelang tahunan or annual rings. Alright, so one ring is 1 year, represents 1 year of growth, and we can estimate the age of the tree by counting the rings. Okay, you can start from the center, this circle here, and count the black lines outwardly, or you can start from outside, count inwards. But actually in the tree, the rings are formed from inside to out. That means in the first year, maybe the stem is only this big, and then second year it becomes bigger and bigger and bigger as it adds on more rings. Okay, the stem becomes bigger and bigger. So let's count the number of rings to know the age of the tree. Starting from this black line here, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay, so this tree is 10 years old. Okay, and each ring is 1 year's growth. Now, describe the formation of these annual rings. So annual rings are formed when the tree starts to undergo secondary growth, which increases the diameter of the plant stem here for the plant to get more support for its leaves. The stem has to grow in size, in diameter. Alright, so this secondary growth occurs a few years after primary growth has occurred. Okay, so just revision. Primary growth occurs at the beginning of the plant's life, okay, from 0 to maybe 3 years or more, and then after that, that's increase in height of the plant. It will grow taller and taller. After that, there's secondary growth together with the primary growth. And the secondary growth is to increase the diameter of the plant stem through formation of the secondary xylem. So all this white part here is the wood. It is actually secondary xylem, which is formed during secondary growth. It's just like any xylem, but it's formed during secondary growth, so it's called secondary xylem. Okay? So primary xylem is formed in primary growth, and secondary xylem is formed in secondary growth. Now, to describe the formation of its annual rings, we have to state the different rate of growth of the plant stem in different seasons. Okay, let's see how to answer this question. P1, secondary growth occurs, okay, in the formation of the annual rings, but at different rates, sometimes fast, sometimes slow, according to the season. Season means spring, summer, autumn, and winter in the temperate areas. but not in Malaysia. We don't have these 4 seasons. So during spring, what happens? The secondary xylem produced in the spring to early summer is larger and brighter due to sufficient sunlight and water supply. And then after that, in late summer, autumn and winter, the secondary xylem formed in the late summer, autumn and winter are darker, smaller and darker due to less light and water supply. Okay, so let's explain this. So here we can see the one ring. This is one ring. Okay, the light wood and the dark wood. Okay, light wood orange color, dark wood is brown color, forms 1 year of growth. So how do we get this light wood and then you get this dark line called the dark wood? Okay, that you can see here. That would tell us that it's 1 year's growth. Okay, now in spring, conditions are favorable. You have a lot of sunlight, water supply, and then the temperature is also higher. So the plant will grow well, and the size of the xylem, secondary xylem, is large like this. Now each structure here is a xylem vessel, so it's large, and then it has a big lumen or hole in the center and thin walls. Okay, so overall when you look from afar, it is bright. Here you say bright, or it is light wood. They call it light wood. Okay, light in color like this. But when late summer, autumn and winter comes, then growth will slow down a bit because there's less light and less water supply. So the type of xylem formed is different. Okay, it is called dark wood. Okay, this brown line here, and it's also thinner. The line, the width of it. So the xylem form here is smaller, has a smaller lumen. The xylem vessels have smaller lumens, and also the wall is thicker. The wall of the xylem is thicker. Okay, so when you look from afar, you can see that it's darker color. Okay, it's called dark wood. All right. So spring, summer, autumn, winter. So that makes up 1 year. Okay, so this is 1 year's growth. Next question here. Diagram 6.1 shows a component of a cell commonly found in human cells, or cellular component. So this cellular component is actually an organelle in the human cell, and we are asked to identify the cellular component shown in this diagram. Okay, so this has been asked before in the earlier question. You can see the folded inner membrane here. So this organelle is the mitochondria, right? Now state the process that takes place in this cellular component. So we know that the mitochondrion is the site of the process called what? Aerobic respiration. Okay, where oxygen is needed for the reaction. All right, so this is the specific term. So if you write cellular respiration, I'm not sure whether you will get your 1 mark, because here they want you to write the word equation for the process. Okay, so you have to clarify here already what is it aerobic or respiration or fermentation. So better to be specific here, aerobic respiration. Now word equation. Think of the acronym, the BIOLOGY acronym called what? GOWAC, right? GOWAC for aerobic respiration. So let's write down glucose plus oxygen, all right, produces carbon dioxide, water, and energy. G for glucose, O for oxygen, W for water, E for energy, and C for carbon dioxide. Simple, right? So use this to remember the word equation for aerobic respiration. Take note, it's a word equation. That means all the words only. Use the words. Don't use the chemical formulas for glucose, oxygen in any part of this word equation. It must be in words. Now next, explain how the substrate used in 6b2 is obtained. Okay, that means the substrate here is glucose. How is it obtained by humans? Right, it has to be eaten, right? So it is found in carbohydrates. And after we have eaten the carbohydrates have to be digested to form the glucose, right? So let's look at the answer. Glucose is obtained through the process of hydrolysis or digestion of carbohydrates such as starch by the enzymes such as amylase and maltase, right? For example, starch is digested or hydrolyzed by amylase to form maltose, and then maltose is hydrolyzed by maltase to form glucose molecules. Okay, and this digestion of food occurs in the alimentary canal, in the digestive system. Okay, so mention the reaction and to produce glucose, and also there is the hydrolysis of carbohydrates, and also where it occurs. Question C. The paddy in watery field in diagram 6.2 below undergoes a different process compared to human cells, right? Name the process that is stated here, right? So we see the paddy is immersed, the roots are immersed in water. So the water will contain very little oxygen, right? So how do the roots carry out the process of respiration without oxygen? Okay, so the process is called alcohol fermentation, right? Where the glucose is broken down into carbon dioxide and ethanol with the release of a little energy, and no oxygen is involved or required. All right. In short, our bio key or acronym or memory aid is GAROCEE or GC. Just now we had GOWAC for aerobic respiration, and now we have alcohol fermentation, and the equation is GC, right? So the alcohol here is actually the ethanol. That's why we call it alcohol fermentation. Fermentation means this type of breakdown of glucose without the use of oxygen, and that there's formation of ethanol in the process. Okay, so take note, glucose breaks down to form carbon dioxide plus ethanol plus energy. All right, and this occurs in the roots of the paddy plant that are submerged in water and have a lack of oxygen. Give one example of another microorganism that carries out the same process as the plant in 6.2. That means also the same equation, and it's also alcohol fermentation. So this microorganism is yeast, right? Now next, state one difference between the processes carried out in human cells and plant cell in the diagram 6.2. All right, so just now we had the human cell that we're talking about, and aerobic respiration. Now we have to compare it with the alcohol fermentation in the plant. All right, so easiest thing is to use the acronyms, the bio key that I've invented. Compare GOWAC with GC, that's all. All right, and give one difference. Right, let's look at the differences. There are actually a few differences, right? Now, firstly, in the human cell, because it says carried out in underline here, human cell and plant cell. So in the human cell, the complete breakdown of glucose occurs. The glucose is broken down completely to form water and carbon dioxide, with the release of this E is for energy, a lot of energy. So in the human cell, complete breakdown of glucose occurs, but in the plant cell, incomplete breakdown of glucose occurs. Okay, so the glucose is also broken down, but is incomplete, so that's why we get the product of ethanol, which still contains a lot of chemical bonds inside. Still a big, comparatively large molecule. All right, and the breakdown of glucose is incomplete here. Okay, for alcohol fermentation. Second point that can be accepted, the process in the human cell occurs in the presence of oxygen. All right, aerobic respiration occurs in the presence of oxygen. But the process in the plant cell, that means alcohol fermentation, occurs in the absence or lack of oxygen, as can be seen here. Next, the process in the human cell occurs in the cytoplasm and mitochondria. So for aerobic respiration, the first part of the reactions, the first few reactions occur in the cytoplasm, okay, which we call glycolysis, right? The reaction is called glycolysis. But later on, the products will move into the mitochondria, where more breakdown occurs, right? And so the whole process of aerobic respiration occurs in the cytoplasm and then the mitochondria, okay, where oxygen will be provided for the other reactions. But the process in the plant cell, that means alcohol fermentation, occurs only in the cytoplasm. Okay, after glycolysis, the products formed are then converted to form the ethanol. And all this occurs in the cytoplasm for alcohol fermentation. There is no mitochondria is involved in the breakdown of the glucose. During alcohol fermentation, mitochondria is not involved. Now, next, the process in the human cell produces water, W for water. But the process in the plant cell does not produce water, right? There's no water produced. In fact, what is produced is ethanol, okay? Instead of water, E here for ethanol. The process in the human cell produces more energy than the process in the plant cell. Okay, this energy produced in the aerobic respiration is much more than for alcohol fermentation, because the breakdown of glucose in alcohol fermentation is incomplete, so less energy is released. So last point, glucose is broken down into carbon dioxide and water in the human cell, right? Carbon dioxide, C for carbon dioxide, W for water. But in the plant cell, for alcohol fermentation, glucose is broken down into carbon dioxide and ethanol instead. All right? So memorize the differences here as they can be asked in the essay questions. A short revision on fermentation. So we know that there are two types of breakdown of glucose. The first type is aerobic respiration, which is the complete breakdown of glucose. Okay? And it requires oxygen. But then you also have the other type, which is called fermentation, the other type of breakdown of glucose. So fermentation is just the opposite of this aerobic respiration. It's the incomplete breakdown of glucose in conditions of limited or no oxygen. And it's actually divided into two types, all right, depending on the organism. The first type occurs in yeast in conditions of lack of oxygen, or in the paddy plants, as we have discussed in the question, that grow in the waterlogged areas with less oxygen. All right, like the paddy plant roots. So for alcohol fermentation, it's the incomplete breakdown of glucose into ethanol, carbon dioxide, and energy. That's the definition. And the equation is GC. G for glucose, C carbon dioxide, E ethanol, E energy. Energy release is 210 kilojoules of energy per mole of glucose, right? Now for the other type of fermentation, it's called lactic acid fermentation. It was not tested in this question, but then it can be asked. So you must know that it is also the incomplete breakdown of glucose, but the product form is not ethanol. It is lactic acid and energy. That's why we call it lactic acid fermentation. The final product is lactic acid. Whereas for alcohol fermentation, the final product is alcohol, which is ethanol. Okay, for lactic acid fermentation, the equation is very simple. Glucose breaks down to release energy, and the lactic acid is formed. So think of what? What is the acronym? GEL. G-E-L. G arrow E-L. Okay, so we have GC here for alcohol fermentation and GEL for lactic acid fermentation. So it occurs in the bacteria lactobacillus, which carries out milk fermentation to produce yogurt. So how's the yogurt produced? You can be asked this question for essay question in the essay section. Now, to produce yogurt, you have to add in the bacteria called lactobacillus into the milk that is prepared, fresh milk that's prepared, and you have to leave it to ferment, right? So the bacterial lactobacillus will carry out lactic acid fermentation and produce the lactic acid. Okay, and then this lactic acid acts on the milk protein and coagulates it. Okay, and that's how we get yogurt. Then the sour taste of the yogurt is due to the lactic acid produced by this bacteria called lactobacillus, all right, from the incomplete breakdown of glucose. Now, lactic acid fermentation is also carried out by the human muscle cells during vigorous training or vigorous exercise, or when the person is running very fast. Okay, so remember these points about fermentation, very important for essay questions too. GC and GEL, remember that. So these are the notes on the differences between aerobic respiration and fermentation. You can read through this and copy it down if you haven't, if you want to. And here I've written here the equation for aerobic respiration is GOEC. For fermentation is GC or GEL. Simple and easy to remember. And then the chemical equations, no, sorry, the word equation is written below here, which can be asked in SPM. As for example, just now you're asked to write the word equation for aerobic respiration. But you can also be asked to write the word equation for alcohol fermentation, which is GC, and lactic acid fermentation, which is gel. Now, this comparison can come out in the essay question. Then, if you're asked to give similarities and differences, you can write down some similarities between aerobic respiration and fermentation. Both processes involve breakdown of glucose, right? The glucose is broken down in both processes, and then it is used to release the energy. And then this energy is used to produce ATP. That's why the energy is converted to chemical energy stored in the bonds of ATP. Okay, and then when you need energy, then the ATP will break down, and the energy release is used for muscle contraction and so forth. So both processes produce chemical energy in the form of the ATP molecules. So the bonds in the ATP molecules store the energy. If the phosphate bond is broken down, then the energy is released. Both processes begin with glycolysis in the cytoplasm, in which glucose is broken down into pyruvate first. All right, but after that, the pathway is different. For aerobic respiration, the pyruvate will move into the mitochondria, where it's further broken down, and oxygen is required for this process. Okay, so that's for aerobic respiration. But for fermentation, the pyruvate continues to be converted to other products in the cytoplasm. So mitochondria is not involved in fermentation. All right. Now, in both types of energy production, the process begins in the cytoplasm. That is, glycolysis occurs in the cytoplasm. Both processes occur in yeast, bacteria, animals, and plants. That means these organisms can choose to carry out aerobic respiration or fermentation. All right. Usually, of course, aerobic respiration is preferable. Unless there's no oxygen available, then the organisms will switch to fermentation to continue to release some energy from the incomplete breakdown of glucose. Right. That's all for this lesson. Thanks for viewing, and stay tuned for further discussions on the state papers. Do share, like, and subscribe. Goodbye for now.